we're going to go through just a very, very brief and basic overview of DNA and RNA. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And these are nucleic acids that are chemical carriers of genetic information in the cell. Basically, they contain information that directs a cell, how its function, and its properties. As you can see in the pictures here, the RNA is a single-stranded nucleic acid, so we have this long chain, whereas DNA is double-stranded, and it makes up what's known as the DNA double helix. And we're going to look more at the structure that makes this up, but what you can see is we have those heterocycles, those nucleobases, the purines and the pyrimidines. Those are attached to the strands, and it's actually the base pairs between the different bases, between the lone pairs and the hydrogens forming hydrogen bonds, that allow for these helix type shapes and allow for the interactions to make up these structures. Here is the sugar ribose, also known as the carbohydrate ribose. Now, these sugars, every carbon has an OH attached on the ring, or CH2OH. But if we lose an oxygen at one of these positions, we end up with a deoxyribose. And it's actually at this 2 position right here, where an oxygen can be lost, and that gives us deoxyribose. And the deoxyribose is the core structure for DNA, and the ribose is the core structure to make up RNA. So what happens from here is we can think of replacing the oxygen on this first carbon with one of those nitrogen bases. So for the purposes of this example, I'm going to replace the oxygen in the ribose with adenine. So now we have one of those bases attached at that position, in this case adenine, but it can be any of them. And this gives us what we call a nucleoside. The second place that gets modified is then this oxygen, the CH2OH over here on the left. What we do is we add a phosphate group to the oxygen. So let's draw that out. Now, once we've added the phosphate, the PO3 group, now we've gone from a nucleoside to a nucleotide. And the components of the nucleotide here in the core, we have the sugar. Right here we have the base. And right here we have the phosphate. And you can have an RNA nucleotide or a DNA nucleotide. The only difference is with a DNA nucleotide, you don't have that OH group at carbon 2. So then, in order to form the nucleic acid, what happens is several of these nucleotides join together. And they join by the oxygen on the sugar with the phosphate on another molecule. So we can kind of schematically write out what that looks like. So here's sort of a simplified diagram of a nucleotide where you have your sugar with the free OH, we have the phosphate and the base, and we can take many of these and join together So let's copy this just a couple of times. Okay, and think about how these might bond together. So what will happen is, to join these together, the oxygen on one sugar will bond with the phosphate of another. So let's just combine those together. Just erase a couple of pieces there. Join. Let's do that again. Another oxygen from this sugar. Bond to there. And keep in mind that these don't have to be the same base. This can be base 1, 2, and 3, and that's what changes from each nucleotide in the sequence. And this gives us our nucleic acid sequence. So that's just a basic overview of how these molecules form the DNA and RNA nucleotides and nucleic acids.